Let us take a look at the fundamental components of prokaryotic DNA replication. Shown here is the way in which prokaryotic DNA is organized. Prokaryotic DNA exists in one circular chromosome. The DNA is still structured as a double helix. In prokaryotes, there is only one origin of replication. Here is where all of replication starts. It ensures that all of the replicon, or segment of DNA that is being replicated, is in fact replicated and only once. At the origin of replication, we see the formation of a replication bubble where the double helix of DNA is unwound and allows for replication of DNA. At either side of the replication bubble are the replication forks. These are the points at which the DNA is being unwound to make the single-stranded DNA template required for DNA replication. An initiator protein binds to the origin of replication and is used to locally unwind a small portion of DNA. This allows the enzyme helicase to bind at the replication forks. Helicase unwinds and unzips the double-stranded DNA molecule by breaking the hydrogen bonds between the DNA nucleotides. Single-strand binding proteins stabilize the DNA by binding to each single strand of DNA. It coats the strands and prevents them from recombining and forming hydrogen bonds again. The enzyme gyrase is a type of topoisomerase. Gyrase's function is to relieve the torsional strain caused by supercoiling, an over- or underwinding of the DNA. Gyrase does this by creating nicks in the DNA ahead of the replication forks. Together, helicase and primase create the primosome. These two enzymes remain at the replication fork. The function of primase is to create a free 3' end required by DNA polymerase, since DNA polymerase can only add to the 3' end of an existing DNA strand. Primase does this by creating a short RNA sequence of about 10 to 12 nucleotide pieces complementary to the DNA sequence called a primer. Focusing on the ends of our DNA, we can determine which strand will be leading and which will be lagging. Because the DNA double helix is anti-parallel, the strands run in different directions, which is why we see the presence of leading and lagging strands. DNA replication occurs in the 5' to 3' direction of the complementary strand, or the strand we are building. This is due to the need for a free 3' end for the DNA polymerase III to add on to. Our leading strand will be built in the 5' to 3' direction toward the replication fork, allowing for continuous replication as the DNA polymerase III can add to the strand as the replication fork opens up. The lagging strand is also built in the 5' to 3' direction, but is built by discontinuous replication away from the replication fork. While the replication fork opens up, DNA polymerase III can only build short segments of DNA in the correct direction before having to start a new segment at the replication fork. These short segments are known as Okazaki fragments. We also see more primers on the lagging strand because of this. The original DNA strand will run 3' to 5' in the direction replication is occurring, whether continuous or discontinuous. DNA polymerase III is the enzyme that, when attached to an RNA primer, adds deoxyribonucleotide triphosphates to the 3' end of the DNA strand. A phosphodiester bond will form between the two nucleotides, cleaving two phosphates in the process. But this is how DNA polymerase III activity synthesizes DNA. Phosphodiester bonds join nucleotides, creating the phosphate sugar backbone of the DNA molecule. DNA polymerase I functions to remove RNA primers, so there is no longer a mixture of RNA and DNA in the new strand. It also replaces the RNA primers with DNA nucleotides. However, a nick, or missing phosphodiester bond, remains between the nucleotides. Ligase's function is to repair the nicks in the new DNA strand by catalyzing the formation of phosphodiester bond without adding another nucleotide, altering the genetic code.